So, good afternoon, everyone. It is my great honor to introduce Professor Juan Bonifacino to give a lecture at our division. Professor Bonifacino is a distinguished investigator at the National Institute of Health, USA, and he is also an associate scientific director in the cell biology branch and the neurobiology branch in the National Institute of Child Health and Human Development at NIH. Professor Bonifacino is a world-renowned scientist in the cell biological field, and he has discovered many influential findings in the protein trafficking area. In the past decades, Professor Bonifacino's group has reviewed a variety of important cargo sorting signals and cargo adapters, and they elucidated the molecular mechanisms underlying how these cargo sorting signals and adapter proteins regulate protein sorting to endosomes and lysosomes. Moreover, Professor Bonifacino has applied these basic cellular molecular mechanisms to elucidate how defects in protein trafficking can cause various human diseases. So without further ado, let's welcome Professor Bonifacino.
Well, thank you very much for being here this afternoon. Good afternoon to all of you. I also want to thank Yu Guo for inviting me to visit this university. I was here for another reason, but I didn't want to miss an opportunity to come here and meet all of you again. Actually, today during lunch, I met with some students who still remember my last visit in 2015, and they remember my talk. But they remember my talk not because of what I presented, but because at the end of my talk, I played a song. So they remember the song. Okay. So today I will not have any music in my talk. So hopefully you will still find it interesting, and you will remember it in a few years when I come back. Okay. So the title of my talk is Coatopathies, Genetic Disorders of Protein Coats, Human Diseases that are caused by mutations in coat proteins involved in protein sorting. Now, before I get into that topic, I would like to give you a very brief introduction to what we are interested in. And as you know very well, the interior of eukaryotic cells is organized into a number of membrane-bound compartments or of organelles that you will be able to identify here: the Golgi complex, lysosomes, mitochondria, secretory granules, and others. And each of these compartments has a specific protein composition which is responsible for their specific functions. And the problem that we and many other people in the field are interested in is how is that heterogeneous distribution of proteins to different organelles achieved? What are the mechanisms that ensure that one protein goes to lysosomes, whereas another protein goes to the plasma membrane? So those are the mechanisms of protein sorting that we are interested in. Now, we are particularly interested in organelles that make up the so-called endomembrane system of cells. So we don't study the nucleus, and we don't study peroxisomes. We study these organelles: the endoplasmic reticulum, the Golgi complex, different types of endosomes and lysosomes, and different domains of the plasma membrane in polarized cells such as epithelial cells or neurons. So how do proteins get to all of those compartments? How are they specifically sorted to those compartments? And one thing that we have learned over the years is that many, and we know that sorting to these compartments is in many cases mediated by so-called protein coats that are associated with specific sites with these organelles where proteins are sorted. And for instance, there are these coats, COP1 and COP2, that function in the early secretory pathway. And then there are 5 other coats named AP1, AP2, AP3, AP4, and AP5 that function in post-Golgi compartments of the endomembrane system. Now, For the remainder of my talk, I'm going to focus on these post-Golgi codes, AP1, 2, 3, 4, and 5. And the function of these codes is to recognize sorting signals in the tails of transmembrane proteins and to concentrate them within these coded areas of the membranes and then promote the formation of a coded vesicle that then transport the selected cargo from one compartment, the donor compartment, to another compartment, the acceptor compartment. Okay, so AP1, 2, 3, 4, and 5, they are 4 subunit complexes that are structurally related to each other. And so the subunits of these complexes are all homologous to each other. These complexes are peripheral membrane complexes that are recruited to different membranes by virtue of interactions with docking factors. So what are those docking factors? They are members of the ARF family of small GTPases or specific phosphoinositides. And the combination of an ARF GTPase and a specific phosphoinositide results in recruitment to different membranes within the cell. Once they bind to the membranes, they can recruit certain protein scaffolds. And for instance, for AP1 and AP2, the scaffold is clathrin. So these are the well-known clathrin codes that are involved in endocytosis and other sorting events. Others have other scaffolds, like AP5 has SPG11 and SPG15, which are clathrin-like proteins. And for others, it's still not entirely clear what the scaffolds are. Then these adapters recognize specific sorting motifs in the tails of transmembrane proteins. And some of them have critical tyrosine residues, and others have leucine-leucine or leucine-isoleucine residues. And essentially what the adapters do is they grab these transmembrane proteins by the tail, and they immobilize them at sites of clathrin, at sites of coated vesicle formation, concentrating them within these transport vesicles. And finally, each of these complexes plays a different role in the cell. Some are very well understood. AP2 is involved in the internalization of endocytic receptors from the cell surface, a transferrin receptor, lysosomal proteins, glutamate receptors, and others. For others, the functions are less clear. AP3 is involved in transport to melanosomes and other lysosome-related organelles, and so on and so forth. They all play different functions, okay? Now, importantly for this talk, mutations in subunits of each of these 5 complexes have now been shown to be the cause of some genetic disorder, some human disease. And I will give you some examples now. For instance, mutations in each of 3 isoforms of the sigma-1 subunit of AP1 cause a different disease. These are neuroepithelial disorders. Some of them result in neurological phenotypes or skin phenotypes or other polarized epithelial cells. And they are known as Metnick syndrome, Fried-Pettigrew syndrome, or susceptibility to pustular psoriasis type 15. Okay? Then mutations in AP4 in the endocytic adapter cause another disease called hypocalciuric hypercalcemia type 3, and that's caused by mutations in the mu2 subunit of AP2. Mutations in AP3 cause the Hermansky-Pudlak syndrome, which is a pigmentation and bleeding disorder. And in fact, mutations in the beta-3A subunit of AP4 were the first coatopathy to be described, and that was in my lab many years ago. And mutations in another subunit of AP3 causes a neurological disease called early onset epileptic encephalopathy type 48. And then mutations in AP4 and AP5 cause a disease that's known as hereditary spastic paraplegia. And it is these mutations in AP4 that I will now like to tell you about. Okay, AP4, one of these 5 complexes. It was first described in our lab and in the lab of Scotty Robinson in 1999. So it's been already 19 years since we published the discovery of this complex. And since then, we've learned, first of all, that it's a component of a non-clathrin coat. Structurally, it's related to AP2, but unlike AP2, it doesn't interact with clathrin. And it's currently not known whether it interacts with a clathrin-like protein. We also know that it's ubiquitous in vertebrates. AP4 is present in every cell in our body. But it's present at low levels in all cells, approximately in the range of 20,000 molecules per cell, which is pretty low number for most proteins. And because of that, it has been very difficult to study. To do immunofluorescence microscopy, immunoelectron microscopy, it's really very hard to study. And that has really slowed down the understanding of the function of this complex. Another limitation is that it's not present in C. elegans or Drosophila, and for that reason we couldn't use those organisms to find out what the function of this complex was. It's an evolutionarily ancient complex though, because it's present in vertebrates, it's present in plants, and it's present in Dictyostelium, and in Aplysia, in Hydra. So it's present in a lot of different organisms. But probably C. elegans, nematodes, and flies lost expression of this complex during evolution. We know that it's associated with the trans-Golgi network, with the TGN. And it's recruited by ARF GTPases to the TGN, that we know. And in addition, we know that the mu4 subunit of this complex binds sorting signals that fit this consensus motif. This is a tyrosine. X is any amino acid. This symbol means a hydrophobic amino acid, leucine, isoleucine, valine, phenylalanine, methionine, any of those 5 hydrophobic amino acids. And E is glutamic acid residue. We know that peptides that fit that consensus motif bind to mu4. And we even know how they bind to mu4 because we solved the crystal structure of a domain from mu4 in complex with a peptide that fits that motif. So we really know a lot about the cellular and biochemical properties of this complex, but we don't know the function, or until recently, we didn't know what it does in the cells or what it does in an organism, what it does in humans. Okay. And part of the reason is that we would knock down the subunits of AP4 in a culture cell, and we would then look for a number of cargos, transmembrane proteins, lysosomes, endosomes, TGN proteins, and everything was the same. Nothing happened. And so we were frustrated for quite some time. But recently, it was found that mutations in any of the 4 subunits of AP4 cause a human disease known as hereditary spastic paraplegia. Types 47, 50, 51, and 52, depending on which subunit of AP4 was mutated. So this was a real breakthrough, because for the first time it suggested that AP4 was important for the development or the function of the central nervous system. So what is hereditary spastic paraplegia? It's a human disease that's caused by axonopathy of corticospinal tracts, neurons that run from the cerebral cortex to the spinal cord and that control the movement of the legs were affected, were sick, and eventually they degenerated, and causing lower limb spasticity and weakness. So essentially stiffness of the legs, right? So that's what hereditary spastic paraplegia means. Now, to date, approximately 100 different genes have been shown to be mutated in human hereditary spastic paraplegia. So it's a very genetically heterogeneous disorder. And the mutations in the 4 subunits of AP4 define a subset of hereditary spastic paraplegia that we refer to as AP4 deficiency syndrome. And it's distinct from other forms of hereditary spastic paraplegia because in addition to lower limb spasticity and weakness, the patients have intellectual disability and they have seizures. And so it's actually worse than other forms of pure hereditary spastic paraplegia. And the anatomical findings in the brain of these patients show that there is axonal dystrophy. So the axons are unhealthy. And there is a thin corpus callosum. That's the structure in the brain that connects the 2 brain hemispheres. And in these patients, it's thinner. Okay? So this is a movie of 2 patients having mutations in the beta-4 subunit of AP4. The first 2 patients with these mutations that were described in the US. And this is a movie of the day that they met at Boston Children's Hospital. So here you can see the typical stiffness of the legs, and she needs a walker to walk around. She's 3 years old in this movie. And this is when they meet. And so they have problems walking. But as you can see, they still have use of their arms and hands. So their arms and hands are less affected by the mutations in AP4. And they are still able to play and to interact with each other. So socially, although they have intellectual disability, they have very poor speech, for instance, they are still able to interact with each other. Mutations in AP4 in hereditary spastic paraplegia have now been found all over the world, mutations in any of the 4 subunits. So it remains a rare disease, but it's present around the world, is pretty widespread. Okay, now, some people think that it may be underdiagnosed as a more common condition, which is known as cerebral palsy, and which is generally caused by a traumatic event during birth. But there is an important, and so the idea is that perhaps as more genetic testing is done, more patients are going to be found to have mutations in AP4. Now, one important difference with cerebral palsy is that AP4 deficiency syndrome is progressive. So these girls and all the patients will get worse with age. It has a neurodegenerative component to it. And eventually, these patients may become paraplegic. So in addition to their legs, they may lose use of their hands and arms, and they may end up in a wheelchair. And this is known from the study of patients that were diagnosed later in life who are in a wheelchair. And so it's a big problem, right? But perhaps early intervention, if we understood the pathogenesis of this disorder, one could intervene early so that one could prevent the neurodegenerative phase of this disease. But in order to come up with a therapeutic approach, we need to understand the pathogenesis of this disorder. How do mutations in AP4 cause this disease? So one approach that we took was to study an AP4 knockout mouse. And this particular mouse has a mutation in the epsilon subunit of AP4. And it was made by deletion of exon 3 and from the epsilon subunit of AP4. And this is a Western blot analysis of samples from the cerebellum and the cerebral cortex from wild type and knockout mice. And as you can see here, the knockout mice have no expression of the epsilon subunit of AP4, so they are true knockouts. And when you knock out one of the subunits of the complex, another subunit also disappears. And that's because when one subunit is missing, the other subunits are degraded by the proteasome, which is something that we showed before. So essentially, these patients have no intact AP4, perhaps a remnant of some of the other subunits. And for instance, the gamma subunit of AP1, another adaptor protein complex, or other adaptor protein complexes are not affected. Only AP4 is affected. Okay, so we obtained these mice. And I see that the movie is not playing, and I'll see if I can make it play. If not, oh yeah, okay. So we obtained these AP4 knockout mouse, and we looked at them, and they seemed fine. Yeah, they were sort of running around. We expected that the knockout mouse would be dragging their hind limbs, perhaps, and they were fine. So for quite some time, we were puzzled and disappointed. We spent all this time making the mouse, and then they seemed to have no phenotype. So, but then we contacted a colleague of ours, a young investigator who had just been recruited to our institute, Claire Lepichon, and she was a real neuroscientist, unlike us. So she studies neurodegeneration in the spinal cord. And so we took the mice to her to see if there was any evidence of degeneration. And the moment she picked up the mice, she said, oh, they have a neurological phenotype. Oh, really? And she said, they have clasping. And so she explained to us that when you pick up in a wild-type mouse by the tail, it will splay the hind limbs in preparation for landing, probably, right? You pick up the mouse and in preparation for landing. But these, the mutant mice kept their hind limbs retracted. And that reflex is called clasping. And she explained to us this is very common of various neurological disorders. It's a rather common, non-specific phenotype of many neurological disorders. But this told us for the first time that these mice did have a neurological problem. So we then started challenging in different ways. And so we conducted a number of motor and behavioral assays. And for instance, one of them is a rotor rod that many of you are probably familiar with. You place the mice on a rotating cylinder, and you increase the speed of the rotation. And then at a certain speed, they fall from the rod, and they activate a sensor. And then you measure the speed at which they fall. And what we found is that the knockout mice fell at lower speed, so they were able to stay on the rotor rod less well than the wild-type mice. So they had a motor defect. And we also performed grip strength tests, and that were not done exactly as depicted here. In fact, we let the mice grab onto a grid using all four limbs. And then we pull from the tail, and you measure the force that takes to detach them from the grid. Some of you are smiling, but yeah, that's how it's done. And we saw that the knockout mice had less grip strength. So they had a number of motor deficits. So although in appearance they seem fine, once you challenge them, then they manifested a series of motor deficits. They also had some behavioral deficits. For instance, they seem to be hyperactive. If you saw that mouse in the picture, it was sort of running around, more so than the wild type. And they also have increased startle acoustic response. If you use a loud noise, then they will be startled much more than a wild-type mouse, but we still don't know how to interpret those behavioral phenotypes. Anyway, we then analyzed the brain. We did histopathology of the brain. And this is a corpus callosum in a wild-type mouse. And these are myelinated axons that run from one brain hemisphere to the other. And as you can see here, the wild-type mouse has a thinner corpus callosum, just like the humans. And in addition, this is a section of the cerebellum of a wild-type mouse and an AP4 knockout mouse, stained with an antibody to calbindin, which stains the Purkinje neurons of the cerebellum, which are these green cells. This is the soma of the Purkinje neurons, and this is the so-called molecular layer, and all the dendrites are pointing in that direction. And this is the so-called granular layer, and the axons go into the granular layer to make synapses with other neurons that are found in the deep cerebellar nuclei. And as you can see here, one difference between the wild type and the knockout was the presence of these so-called spheroids, or swellings of the axons of the Purkinje neurons, which were much more numerous in the deep cerebellar nuclei. So near in the distal axon, there were many of these spheroids or swellings that were not present in the wild type. So this is the axonal dystrophy that is similar to that has been seen in the human patients. So from all of these experiments, we concluded, first of all, that the AP4 knockout mouse is a good animal model for AP4 deficiency syndrome. And if one wanted to test certain therapies, so the mice could be used for that purpose. In addition, it demonstrated that AP4 is important for the development and function of the central nervous system. So before we didn't know what it did, now we know that it's really important for the brain. But we still didn't answer the question, what does it do at the cellular level? We now know it's important in the brain, but what does it do at the cellular level? And we thought that the key to answering this question was to identify specific cargos for AP4. Because that's what adapters do. That's what AP1, 2, 3, 5 do. They sort cargos, transmembrane proteins. So what are the cargos that are specific for AP4 that might be affected in this disease? So in order to do that, we performed a tandem affinity purification and mass spectrometry analysis of cells that were expressing the epsilon subunit of AP4 with two tags. Right? We purified the epsilon subunit, and then we did mass spec. And these are the top hits in this purification. The 4 subunits of AP4, that's good. So the affinity purification really worked. Then another protein called tepsin, which is an epsin-like protein that is specific for AP4, and which had been previously identified by the group of Scotty Robinson, and that we also did some work on. So that was great. Then there is this other protein, FAM160A1, which is a hook interacting protein that we are currently studying for its role in AP4-dependent sorting. But the top transmembrane protein, and therefore putative cargo for AP4, was ATG9A, or autophagy-related protein A. So we decided to test whether ATG9A was indeed a cargo for AP4. So the first thing that we did was to confirm the interaction of ATG9A with AP4, and we did it in various ways. This is ATG9A. It's the only transmembrane component of the core autophagy machinery. All the others are cytosolic proteins that are recruited to membranes, but this is a transmembrane component. It has 6 transmembrane domains and an N and a C-terminal tails. Now, interestingly, the amino terminal tail has this sequence, YQRLE, which fits the consensus motif for binding to the mu4 subunit of AP4 that we had previously defined for another protein. So at this point, I wonder, why didn't we find this before bioinformatically? How didn't we find that ATG9A had this motif? And I have no explanation for that. For some reason, we missed it. But now we found it by affinity purification. So we decided to test whether this amino terminal tail interacts with mu4, as would be predicted. And that was indeed the case. We performed a yeast-to hybrid analysis of the amino terminal tail of ATG9 against the mu subunits of 4 complexes. And we found that it interacted specifically with mu4 and not with the other mu subunits of the other AP complexes. And we also performed an affinity purification and Western blot analysis with ATG9A GFP, and we found that this construct was able to pull down the beta-4 subunit of AP4, but GFP wasn't. And we then mutated each of these residues to alanine to see whether that was the motif that interacted with AP4. And we found that the tyrosine and the glutamine were important. The arginine was not, and the leucine and glutamic acid residue were important. So indeed, this motif was responsible for the interaction with mu4. And also, this pull-down assay showed that ATG9A GFP can pull down endogenous AP4, but if we mutate just the tyrosine residue, we lose that interaction. So this is not just any kind of accidental pull down or anything. It's a very specific interaction that is mediated in ways that we understand very well, even structurally. Okay, so they interact, but what is the impact of knocking out AP4 on the sorting of ATG9? So that's the next question that we addressed. And in order to do that, we studied several AP4 epsilon knockout lines. One was HeLa cells, the other was HAP1 cells, which is a leukemic cell line, and then the mouse embryonic fibroblasts from the mice that I just showed you. And as you can see here, all the knockouts don't express any AP4 epsilon in comparison to their wild-type counterparts. Now, one interesting observation that we made is that when we blotted for endogenous ATG9A, there was an increase in the levels of ATG9A in all the cell lines. The fold increase varied, but in all of them, there was more ATG9. So that already told us that something was going on in those cells with ATG9. And then we looked by immunofluorescence at the distribution of ATG9A in wild type and knockout cells. And this is what we saw. This is endogenous protein, and it's important to do these experiments for the endogenous ATG9. In wild-type MEFs, ATG9 is distributed throughout the cytoplasm. Part of it is at the TGN, as you can see here, but then there are these peripheral structures, which are either pre-autophagosomal structures or endosomes, as previously shown by others. And this distribution reflects the fact that this protein cycles between the TGN and peripheral structures on and in forward and retrograde manner. And we then looked at the distribution of ATG9 in AP4 knockout cells, and this is what we saw. Wow. So when they called me to the microscope to show me the results, I said, wow, this is amazing. It's clear, one of the most clear mis-sorting phenotypes that I've seen for any of the adapters. Because in the knockout cells, ATG9 was concentrated at the trans-Golgi network and depleted from the periphery of the cells. So this is in mouse embryonic fibroblasts. This is a normal human skin fibroblast. And again, the distribution of ATG9 is similar. And this is a skin fibroblast from a patient with AP4 deficiency syndrome that has a mutation in the mu4 subunit of AP4. It's also concentrated at the TGN and depleted from the periphery. This is a This is a mouse hippocampal neuron from a wild-type mouse. And in green is ATG9. And as you can see, it's concentrated at the TGN in the soma of the neurons. But it's also found in the neurites. And although here we didn't distinguish dendrites and axons, I can tell you that it's both in dendrites and axons. So ATG9A is not polarized. It's found throughout the neuron. And in the hippocampal neurons from the AP4 knockout mouse, it was all concentrated in the soma and depleted from both the dendrites and the axon. So the results of these and many other experiments, I could keep showing you sections of the brain, the cerebellum, the spinal cord, everywhere. ATG9 was missorted in the absence of AP4. So from these experiments, we concluded that AP4 is required to sort ATG9 from the trans-Golgi network to the periphery, probably pre-autophagosomal structures. Okay, so what does ATG9 do? What is the impact of ATG9A missorting on autophagy, which is what it's involved in? And so you are probably very familiar with autophagy, so I will just point out some things that are important for this discussion. Autophagy is a process by which cytoplasmic particles, organelles, damaged proteins are engulfed within a phagophore that eventually closes and then fuses with the lysosome to form an autolysosome, and where the autophagic cargoes are degraded. Okay, and ATG9A is believed to leave the trans-Golgi network in a specific type of vesicle that is known the ATG9 vesicle, and which we now know is formed in an AP4-dependent manner, and then to interact with the forming autophagosome with the phagophore, perhaps delivering lipids to the growing autophagosome. And this delivery of lipids is required for another autophagic protein known as LC31 to be lipidated and then associated with the membrane of the autophagosome, promoting the capture of cargo and eventually the closing of the autophagosomal membrane. And previous work has shown that ATG9 is required or promotes this conversion of LC31 to LC32. So we decided to analyze what happens with this conversion in the AP4 knockout cells. Initially, this was done in HeLa cells. And this is what happens. You can analyze LC31 and LC32 by Western blotting. And as you can see here, the ratio of LC32, the lipidated form, to LC31, the soluble form, was altered. It was decreased, approximately twofold, threefold in other cell lines. So there was indeed a defect in the conversion of LC31 to LC32 in the lipidation of LC3. But you may also notice that there was another difference. The total amount of LC3 went up. So although the conversion was less efficient, the total amount of LC3 went up by 2 to 3-fold in different cell lines. So, and we think that this is a manifestation of a compensatory mechanism by which cells that are unable to deliver enough ATG9 to the growing autophagosome, what they do is they upregulate ATG9, as I showed in a previous slide, and they also upregulate LC3, so that in the end, the amounts of the active form of LC3, LC32 here, are practically unchanged. And this is something very interesting. We think that cells, mice, and humans with mutation in AP4 partially compensate for the inefficiency in ATG9 transport by making more ATG9 and more LC3. And for that reason, I think the patients are alive because that compensation has taken place. So this is to put it schematically. So AP4 then mediates sorting of ATG9 from the TGN to pre-autophagosomal structures, and that promotes the conversion of LC31 to LC32. If you don't have AP4, then ATG9 gets stuck at the TGN, and a smaller amount reaches the pre-autophagosomal structure, and that decreases the conversion of LC31 to LC32. But the cells react by making a lot more ATG9 so that enough escapes the TGN, even in the absence of AP4. And they also make more LC31, so that in the end, you have almost normal concentrations of LC32, thereby maintaining a basal level of autophagy. And that can be seen in this immunofluorescence microscopy analysis. So this is a staining for endogenous LC3 under basal conditions in mouse embryonic fibroblasts. These are wild type, and these are AP4 knockouts. So the green dots are LC32 that's associated with the forming autophagosomes and with autophagosomes. As you can see here, there isn't much of a difference. And if you now starve the cells of amino acids, then autophagy is induced, and there is more LC32 that's associated with autophagosomes in the wild-type cells. But the AP4 knockout cells also are able to activate autophagy and to increase the levels of LC32 that are associated with autophagosomes. However, we noticed some differences, though. Although they were able to associate more LC32 with autophagosomes, we noticed that the size of these LC3 structures are increased in the AP4 knockout mouse. So although they are able to form autophagosomes, it appears that those autophagosomes are not completely normal and have an aberrant structure, which is something that has been seen before in ATG9 knockout cells. Okay, so we also looked in neurons, right? Because that's where the defect in AP4 are manifested as a neurological phenotype. And we found also that the basal levels of LC32 in neurons were not any different in wild type and in knockout mice. And we also tried starvation of the neurons, but we failed because starvation is not a response that you see in neurons. Neurons do not respond to starvation with increased autophagy because that's not what their physiology dictates. And so one process for which neurons use autophagy is the degradation of damaged proteins or aggregates in the axon. And in fact, it's been shown before that if you have mutations in the autophagy machinery, then neurons are not able to handle aggregates very well, and that results in axonal degeneration and eventually neuronal death. So we decided to test if the AP4 knockout neurons were able to handle an abnormal aggregate in the axon, like the wild-type counterparts. So we decided to overexpress an aggregation-prone mutant of Huntingtin with an expanded polyglutamine repeat. So Huntingtin is a protein that has an expanded polyglutamine repeat, CAG repeats that encode polyglutamine. And normally, we have Huntingtin with up to 36 copies of CAG or glutamine, and that's fine. That's normal, and we are okay. But if this polyglutamine repeat is expanded to greater than 36, then there is an increased probability that the patients will develop Huntington's disease. And so what we did, and that's because those Huntingtin proteins with an expanded polyglutamine repeat tend to aggregate in the axon, and that causes axonal pathology. So we decided, and this has already been shown, that overexpression of an aggregation-prone mutant of Huntingtin results in axonal degeneration. So we decided to overexpress a mutant huntingtin with 103 glutamine residues, and that results in a protein that has a great tendency to aggregate. And in wild-type cells, we found that overexpression of this huntingtin mutant resulted in the appearance of some aggregates in the axon. This is the axon of this neuron. And you can see these aggregates that colocalize with LC3. So they are probably aggregates that are being engulfed by the autophagosomes in the normal cells. And in the AP4 knockout cells, we found higher levels of the aggregation-prone huntingtin and a larger number of these aggregates in the axons of these hippocampal neurons, which also colocalize with LC3, meaning that they were autophagosomes. And this was quantified. And we found that there was approximately a twofold increase in the number of axonal foci that accumulated this aggregation-prone huntingtin protein. So it appears that the AP4 knockout neurons are able to keep a normal basal level of autophagy, but when you challenge them by overexpression of an aggregation-prone protein, they have a decreased ability to degrade those aggregates. And we think that that might contribute to the neuropathology that is seen in this disease. And with this, I want to summarize the conclusions of this work. So I have told you about coatopathies, genetic diseases of protein coats. They are multi-system disorders. Depending on which AP complex is affected, you will have defects in different organs. But a common feature of most of these coatopathies are defects in the central nervous system. And the reason why the CNS is more susceptible to mutations in adaptor protein complexes is probably because of the polarized structure and the large size of the axonal domains, so that neurons are much more dependent on trafficking than other neurons. And that's why the first neurons that are affected by mutations in adaptor protein complexes are neurons with very long axons, such as the motor neurons that go from the cortex to the spinal cord, or from the spinal cord to the limbs. Now, I've also shown that AP4 knockout mice exhibit motor deficits and a thin corpus callosum, axonal dystrophy, which are all features of the human patients. So these mice are good models for the human disease. And we didn't know anything about the function of AP4, the physiological function of AP4. And we now know that AP4 mediates signal-dependent export of ATG9 from the trans-Golgi network. So we are very pleased after so many years of having a very well documented function. And the AP4 knockout cells exhibit impaired LC3B lipidation, but with compensatory increases in ATG9 and LC3, which to a great extent compensate for the absence of AP4. But in the neurons, when you challenge them by overexpressing an aggregation-prone protein, then that results in increased accumulation of the aggregates, and in other phenotypes that I didn't have time to, or maybe I would have had time to describe to you. So we think that defective autophagy under conditions of neuronal stress may contribute to the pathogenesis of AP4 deficiency syndrome. So we think that AP4 deficiency syndrome is another type of diseases caused by dysfunction of the autophagy machinery, and there are a few others that have been shown to be caused by mutations in components of the autophagy machinery. So this is another version of those so-called congenital disorders of autophagy. And so there are mutations in other components of the autophagy machinery in humans or in mice that result in axonal dystrophy, a thin corpus callosum, and motor and behavioral defects. So we think that AP4 deficiency syndrome is related to those other diseases. And with this, I want to end by acknowledging and thanking the people in my lab who did this work, mainly Rafael Matera, Rafaela De Pache, Sang-Yun Park, Ariane Foster, Jeff Mercuria, and Charlie Guardia. And also our collaborators. These studies took a long time, really, to come to fruition. And so Miguel Skrzewski did some of the behavioral assays, and that was something that we couldn't have done ourselves. And Markus Damme in Germany did some of the histological analysis, which I didn't have time to show you. And I also want to thank my institution, the NIH, for funding this research, and you for your attention.
It's truly a wonderful talk. So initially, you briefly mentioned this AP4 has very low molecular number in the cell. Do you think that can be reconciled to its function in regulating the autophagy?
Yeah, I think so.
The later
I think
is
so.
important.
So yeah, it's present at very low level. Unlike, for instance, AP2 is very abundant, and AP1 is very abundant as well. And whereas AP2 and AP1 have many, many cargoes. I mean, AP2 mediates internalization of many receptors. I think that AP4 is going to mediate sorting of a much smaller number of cargoes. And so perhaps you don't need a lot of AP4 to sort those cargoes. So that's one possible interpretation for how such a low level is still important, right, for physiology. So that's my interpretation. Also, in mice, if you knock out the subunits of AP1 or AP2, those mutations are embryonic lethal. In the case of AP4, they are not lethal, although the humans and the mice have problems. So again, that's consistent with the fact that they are adapters for a much smaller set of cargoes, and we think that the cargoes are going to be very specific. So we don't think that ATG9 will be the only cargo. There will probably be others. And there is work from the Robinson lab suggesting that two other proteins called serine1 and serine3 are also cargoes for AP4, but there will be a small set of specific cargoes for AP4. So you don't need very high numbers of this adapter in the cells.
Oh, thank you for a nice talk. So you do see a defect of the fourth generation of the muscle, and you explain it's from the CNS defect. So I just wonder if you see some difference of the sarcomere structure or the fiber size in the muscle.
No, nothing evident, nothing obvious. We did a histological analysis of the whole mouse, and except for the thin corpus callosum and the axonal spheroids, we didn't see other abnormalities. But perhaps we didn't look, carefully enough for very specific phenotypes. But we don't have any evidence for any other defect. Okay, that's a great question. We looked at, from birth to 8 months of age, and we didn't see a progression in the phenotype. At 3 months and 8 months, the motor tests and behavioral tests gave similar results. And the spheroids were present at 3 months, and they were there at 8 months. Now, we haven't done even older mice. So at present, we don't have evidence that in the mice the disease is progressive as it is in the humans.
So as AP4 is ubiquitously expressed in all cell types, so why knock out these genes, or the brain is the most vulnerable part?
Yeah, that's always the question, right? There are many examples of mutations of proteins that are expressed in all cells, and yet the most evident phenotypes are neurological. And again, it's probably because neurons are more dependent on trafficking than other cells. In other cells, things may not have to go very far, so there may be alternative mechanisms. In neurons, with these very long axons, you need trafficking to be working perfectly. I forget the percentages, but I heard that somebody was telling me that more than 50% of all mutations have a neurological phenotype and in humans. So anyway.
One great talk. So you mentioned that in flies and C. elegans do not have AP4. So if you go back to look at the ATG9 in these two organisms, do they correlate?
That's a great question, and that's a problem. I don't have a good answer for that. So Saccharomyces, yeast doesn't have AP4 either. Yeast, C. elegans, Drosophila, they have ATG9, but they don't have AP4. So how do they sort ATG9 out of the TGN? It's an interesting question, and I think some C. elegans people are looking for that now. In yeast, there are two other autophagy proteins. I think one is called ATG23, and there is another one that I forget the number. And those are yeast-specific autophagy proteins that are not present in any other organism, and they are required for sorting of ATG9 out of the TGN. So maybe other organisms have evolved other proteins to export ATG9 from the TGN, but we don't know. You ask all the right questions. Yes, and that's a problem for our explanation because they have the tyrosine motif. It's a little bit more degenerate, but the tyrosine motif is there, but AP4 is not. So we don't have a good explanation for that. In yeast, on the other hand, ATG9 doesn't have the tyrosine motif, and it uses two other proteins to get out of the TGN. But in C. elegans and Drosophila, there is a tyrosine motif there. And in humans, there are two ATG9s, A and B. A is ubiquitous, and B is expressed only in placenta and in neuroendocrine cells. And the N-terminus of ATG9B is very different from ATG9A, but the motif is conserved. So although we haven't looked at ATG9B, our prediction is that it will also require AP4 for sorting. So that motif is critical because the rest of the tail can be totally different, but the motif is conserved. And the motif is conserved in all other organisms except for yeast. Yeah.
Okay, so it's a very wonderful talk, actually. I have one question about the specificity. So basically, you said that this AP5 is related to autophagy, and autophagy is a very important pathway to remove the aggregated protein in the neuron. But why, you have special, problem in the corpus callosum, not, hippocampus or cortex or striatum, which play a very important role in Huntington or Alzheimer?
Yeah, no, that's a great question. I think that defects in the corpus callosum are easier to see and than in other parts of the brain because you have these long axons crossing the two hemispheres, right? So in our experience, whenever there is a defect in axon, it's easier to see in the corpus callosum than in other parts of the brain. And I think that suggests that there is a problem with either axon growth or guidance or maintenance of these myelinated axons in the corpus callosum. And we don't know what exactly is affected. But there is another group at University College in London, I believe, Joseph Kittler's group, that is studying the AP4 knockout mice. And they see that there is a defect in axon growth. Axons grow less well in the AP4 knockout mice. And so perhaps the thinner corpus callosum reflects a problem with the extension, the growth of those axons.
Here. So in the AP4 knockout neck cell, the ATG9 stock in the Golgi is very striking, right? But the autophagy phenotype is relatively mild. So where did the compensation come in? Because more proteins cannot explain. If you have more ATG9s, their stock then it cannot.
Yeah. Well, I think that that's a great question, and I think that, let's say, if you have AP4, then the export from the TGN is sufficient. Maybe you export, 90% goes in AP4 coated vesicle. If you don't have AP4, there may still be some leakage because you are expressing more. Enough is leaking out of the TGN non-specifically to support some level of autophagy. Like, for instance, if you knock down or out AP2, the endocytic adapter, the transferrin receptor, which is a cargo for AP2, some of it will still be internalized by the normal turnover of the plasma membrane, a much smaller fraction, but it will still be internalized, and probably the same thing happens at the TGN. So that's our interpretation.
Very nice talk. So I noticed the majority of the data was generated by the cell lines, like the cancer cell lines. I'm wondering, any data was showing the neural cells, and do you think the AP4 deficiency may affect the peripheral delivery of the ATG9A into the axonal terminal, especially like you say it's linked with the syndrome. So I'm thinking if they are in the same level in the brain neurons and also the motor neurons in the spinal cord.
Yeah, that's a great question. It is something that we are beginning to look at. Autophagy in the axon works, and this was studied by people like Erika Holzbauer and others. So autophagosomes in the axon appear to form in the distal axon, in the axon terminal. And that's where you see LC3 positive autophagosomes form if you do live cell imaging. And then they start moving in a retrograde manner in the axon until they probably encounter lysosomes that are coming from the soma, and they fuse and they form an autolysosome, and that's how they degrade things in the axon. And what we have seen, and probably we think that ATG9 vesicles are also coming from the soma and delivering lipids or whatever they do to the forming autophagosomes in the distal axon. One very interesting observation that we made is that in the AP4 knockout neurons, the LC3 positive autophagosomes form in the distal axon, but they cannot move in retrograde manner, and they continue to accumulate there. So it appears that the fusion or the delivery of whatever ATG9 delivers to the forming autophagosome is required for the autophagosomes to mature and then to begin to move in retrograde manner. So that's another phenotype that we have seen in the neurons of the AP4 knockout mice. The LC3 positive autophagosomes accumulate in the distal axon, and they are relatively immobile.
Okay, so if no questions, so let's thanks Professor BONIFACINO again.
Thank you very much.
Thank you.